Thank you, Bibi. I think we can get started then. So, hello, everybody. My name is Dominic McGurr, and I'd like to welcome you on behalf of Basel Biometric Society to today's webinar on controlling the chances of false discoveries in exploratory analysis of clinical trials. The organisers, Kostas Sakidis and Frank Bretz, and Bibiana Blattner have put together a very exciting event. I think this is reflected in a very high number of registered participants, almost 500 registrations. And as one might expect for such an event, most of you are coming from a statistics background. It's always nice to see participants from other areas of expertise. So we have large numbers today with clinical, epidemiology, health economics, and regulatory backgrounds. It's also always impressive to see the global reach of the Basel Biometric Society. Today we have people joining from all regions of the world and 37 countries in total. Just a few pieces of housekeeping. This event will be recorded. In fact, it already is being recorded, and the recording and presentation slides will be made available, and you can find them on the Basel Biometric Society after the event. We'd like to ask you to please stay muted unless you are speaking. If you are one of the speakers, please also make sure to unmute yourself before you start your presentation. We will have time for Q&A at the end of each presentation. We'd like to ask you to please put your questions in the chat during the presentation. There is the possibility to raise your hand during the Q&A, but the chat is preferred. And in case you have any problems during the event, please contact Bibiana, and you see the email address on the screen here. Today's webinar is on the topic of multiple test problems, and therefore I'd like to take the opportunity to link back to a previous event from the Basel Biometric Society, the 2022 training on graphical approaches to multiple test problems from Eckhard, Glim, Frank Bretz, and Zhongji. And all the recordings, all the materials from this event can be found on the website, along with many other past seminars and past trainings. And while the event from 2022 was focusing more on traditional methods for multiple testing, which are very well established in drug development, today we're going to hear about more recent advances in multiplicity control. And turning to the agenda, Kostas and Frank have put together a very impressive lineup of speakers. The webinar today will be split into two sessions. So part one will be about controlled variable selection. We'll have a 15-minute break, and part two will be on controlled subgroup discovery. And since Kostas himself is the first speaker and he's sitting in the room with me, I have the pleasure to introduce him. Kostas Sakidis is a member of the Advanced Methodology and Data Science group at Novartis. He specializes in exploratory analysis for biomarker discovery, digital biomarker development, and assessing treatment effect heterogeneity. He earned his PhD in machine learning from the University of Manchester, and before joining Novartis, he was a postdoctoral researcher at AstraZeneca and Roche. Please, Kostas, go ahead.
Thank you very much, Dominic, and thanks to the BBS Basel Biometric Society for giving us the platform to organize this event. So in this first presentation, we'll talk about some works that we are doing on using some recently suggested framework from academia, namely knockoffs, to control type 1 error when we want to explore prognostic and predictive variables in our clinical trial programs. So a bit of the agenda in high level. Firstly, we'll discuss what we mean when we say variable selection via machine learning. Then we'll touch the point of the quantification of uncertainty via knockoffs. And then we'll show how we took this framework, we adapted a bit to target the predictive biomarkers, which is of interest to our trials. And then I will have a case study from the psoriatic arthritis. I need to emphasize this joint work with many great collaborators and co-authors. And currently we have these three papers around this set of research. And also there is a R repo with the name knockoff tools. So if you use it and you have any feedback, feel free to let us know. Now let's set up a bit the scene. We call variable learning, machine learning, feature selection. And more particularly, we will focus on what we say supervised feature selection, where we have a response variable y, which is our target variable, and can be continuous like the disease duration or the status, binary, healthy or unhealthy, let's say. And then each patient, each person can be characterized with a large number of other features. Can be gene expression data, or can be measurements collected by digital sensors, or it can be simply the clinical variables that we have at the baseline that they describe the patients. And in most of these scenarios, it's safe to assume that only a subset of variables influence the outcome. And this is the target of our work. We want to identify which variables influence the outcome, and this we can use it as a starting point to answer many interesting questions. And these variables, we call them also prognostic biomarkers. Now a bit of annotation. We have x1 to xp. The columns are the different variables, y is the target, and we define a variable to be relevant if the probability of observing the target given this variable and all of the rest is different than observing the target given all of the other variables. So in other words, this variable contains some unique information towards the target that is not captured by the rest of the variables. And the set of these relevant variables, as tilde, this the purple one that I have here, it's the optimal set and what we are going to extract. And if we have this and condition over this, all of the rest of the variables are irrelevant or independent of the target variable. So now let's imagine that we have one scenario that we know the ground truth, that the 1, 4, 6, and p variable are relevant, and we have some methodology that estimates the relevant set as these variables plus x2. So the x2 we define as false discovery. And in this selection, the proportion of false discoveries is 1 out of 5, 20%, which means that if we move on and we explore all of this selection, we can manage that it's biomarkers and we want our lab scientists to explore all of them if they are promising or not, we'll waste 20% of our time and our resources to things that will not be replicated at the end. So having some methodologies that they control some sort of a measure of false discovery, it's quite critical. Now, how we do the variable selection in machine learning? We have this training data set. We take all of this, proper strategies, cross-validation, and so on, and we have at the end a machine learning model. Which here we have this black box. Now there is a big area of research in machine learning community of explaining these models, and a critical part is some global scores of variable importance. And there are many ways of deriving variable importance scores, which are model dependent, but also we have model agnostics. And Dennis in the following presentation will discuss about this. But let's imagine now we have, for example, the Lasso model. This by design enforces sorts of sparsity. It sets the weights of some variables to the coefficients of some variables to 0, so they don't influence the y, and then give some different scores to the others. Or we have models like the graded boosted trees or the random forest that they don't enforce this sparsity, but again, there are multiple ways of deriving importance scores to understand which are, let's say, the more important variables inside this model. And the same can happen with deep neural networks. And as I said now, almost with all of the models, there are ways of explaining it, and also there are model agnostic ways. Now, a typical activity of a data scientist that we build the model, and this is some example where the variables are blood measures, and we rank the variables according to their importance. So the top is the more important variables, and then we go down. But then we end up with the question, okay, how will we choose the set? What is the most relevant variables in this ranking? And there is some approaches. For example, we can always select a fix, or we can use another validation set to identify the optimal number of top variables. But these approaches, maybe they don't answer questions that were more interesting. So for example, as I mentioned earlier, maybe a more meaningful selection would be the one that will provide some guarantees over controlling the expected proportion of false discoveries, false discovery rate, or controlling the expected number of false discoveries, the per family error rate, or controlling the probability of making at least one false discovery, the family-wise error rate. And I'm sure the ones with most stats background, you have come across all of these terms, and actually it's a very active area of literature under the multiplicity or multiple hypothesis testing, that suggest a lot of methodologies of controlling this error when we perform many hypothesis tests. So when we have, simply speaking, a lot of p-values and we want to control some type 1 error. Now, in the left side, we have importance scores. We don't have p-values. So the main message of this work is how we can act on top of the importance scores that the machine learning model returns and control some measure that we have in the right. Now I will keep motivating a bit the work with some simulations. So in this simple scenario, I generate 500 patients, and each patient is characterized by 200 variables features. And only the first 51 is the ones that are relevant. And then we generate the data set and we fit a lasso model. As I said, here in the x-axis, it's the features. The first 50, the blue, are the relevant. The red are the null, the noise. And to the y-axis, we have the importance. In this case, it's the absolute value of coefficient of the lasso. Now, as I said, lasso enforces some sparsity, so many things are set to 0, but there are other things that are non-zero, and in practice, many times we move with this as the selections. In this case, there are 4 things that can happen. The model correctly identified 32 variables as relevant, gave non-zero coefficient while they were blue. 18 variables, where they were relevant, the model set them to 0, so we missed the selections. Then 117 variables correctly didn't select it because they were irrelevant. But the thing that is more that worries us is that 33 variables, which was noise, irrelevant, they have been assigned a non-zero coefficient. So if we move with these selections, the false discovery proportion in this case is almost 1 out of 2. So 1 out of 2 of our findings are noise. They are not inside the set of variables that we are interested in. And I want to emphasize this is not an issue with lasso. We can have the same with random forest. Okay, this time the importance score is this permutation-based importance. So we have most of the time non-zero coefficient, but we select using this selection. We select 40 variables, and in this selection we have almost 1 out of 3 are false discoveries. The false discovery proportion is almost 1 out of 3. And a bit of a background, in this seminal paper, Benjamini and Hochberg, back in 1995, they introduced also the concept of the false discovery rate, the expected value of false discovery proportion, and they suggested some novel methodologies of controlling the false discovery rate, which, as I said, always it relies on top of p-values, and was making some further assumptions, for example, over independence of the p-values. In machine learning, while maybe there is ways of deriving p-values under some models or with some sort of asymptotic theory, always is a bit questionable the quality of these p-values. So the idea was, can we control this type of error using the importance scores? And actually, a bit more than 20 years afterward, the group of Emmanuel Candes in Stanford published this nice work that they were introducing this concept of knockoffs. And in this slide, I will try to give a very high-level idea what it is. It's the knockoff procedures. It can be seen as a 3-step procedure. In the first step, we construct some fake variables, knockoffs that we call them. Then in the second step, we fit the original and the knockoffs in the machine learning model, and we try to understand, let's say, how more important the original looks than the knockoff. And the third, we have some methodology of thresholding these knockoff statistics that we derive in the second step and controlling the FDR in the nominal level that the user wants. And a few years later, they had also follow-up work that they were showing how we can use this machinery to control the family-wise error rate or the per family error rate. So the 3 error measures that I talked earlier. So in this slide, I will try to go a bit in more detail, but without getting too technical. As I said, the first step is to construct the fake variables, the knockoffs. This is, in machine learning terminology, we can see it as an unsupervised step. We don't use the Ys to generate the knockoffs. And the most important property that we want the knockoffs to obey, it's what we call exchangeability. So the joint distribution of the original features and the knockoffs remains the same if we swap original with knockoff features. Now in this visualization, I just swap 2. You can imagine 1, 2, 3. In these swaps, the joint distribution remains the same. And you understand that this is very difficult property. So generating knockoffs that they obey this property, it's very challenging, and it's the core of these methodologies. And it's very active area of research because in statistics and machine learning community. So in the very first paper, they were using some sort of assumption around multivariate Gaussian. Then they had work that they were relaxing by assuming some hidden Markov model. And then there were various work around deep learning and many other ideas to generate valid knockoffs. Now, in the second step, we take the original features and the knockoffs, and we fit the machine learning model in this space of the original and the knockoffs. And then we calculate importance scores for both of them. And in the second step, in the original paper, they introduce many knockoff statistics. So, for example, from the random forest, it can be the absolute value of the importance score of the original feature minus the absolute value of the importance score of the knockoffs. Or in the lasso that we saw, we can be the absolute value of the coefficient of the original minus the absolute value of the coefficient of the knockoff. The core property in this step for the knockoff statistic is antisymmetric, which means if I swap the original feature with the knockoffs, the only thing that changes in the knockoff statistic is the sign. The magnitude is the same. And then we go to the third step, which we calculate a threshold to control a false discovery rate. And I need here to emphasize that before running this analysis, the user has pre-specified a level of false discovery rate that he wants to have in the selection. So according to this, in this example, let's say the user was this 30%. And in the third step, what we are doing, we rank the features according to the absolute value of the knockoff statistic. And with plus blue, it's the ones that they have positive knockoff statistic. With minus red, it's the one that they have negative. And then we use a threshold. Let's say that we use this tau. Because of the properties of how we generate the knockoffs and how we calculate the knockoff statistic, they prove a very powerful lemma in their papers that they show that we can estimate the false discovery proportion by this expression. And actually, it is 1 plus all of the minuses in this area divided by all of the pluses. So in this case, we have 1 plus 1, 2 divided by 4, 50%. And then we start moving towards the left threshold till the point that we are below of the nominal level of the user. Then we return the feature with a positive knockoff statistic, and this is provable, controls the false discovery rate to the level that the user wants. So this was sort of in a nutshell the presentation of the knockoffs. Now, how we use it in clinical trial. One thing is that the vast majority, not to say all of the papers, in the beginning was focusing when the axis, the input space, is continuous variables. But in clinical trials, we have most of the time we have a mixed variables. We can have, variables like the ethnicity, the gender, and then we have continuous markers from blood. So with this work, Matthias Cormackson and the colleagues, they suggested a methodology of generating knockoffs that can handle these mixed data types. And then we had a follow-up paper that now we didn't focus in this identifying the prognostic variables, but to see how we can switch this framework to target the predictive variables. And because maybe in the audience they are not familiarized with this slide, I'll try to explain what we mean predictive variables or predictive biomarkers. The case study that I use, it's one from the non-small cell lung cancer, and this, it's a famous case study, and the scientists knew that there is some, because of the mechanism of the drug, was targeting people with positive EGFR mutation in the tumor. So the people with positive EGFR mutation, they respond better to one treatment, while with negative to the other one. So now the EGFR mutation, it doesn't tell me something for the why, but it tells me something about the treatment effect. So with our work, the main question of our work was, can we have a mechanism to identify this type of variables that tell us something about the treatment effect, and at the same time control the chances of type 1 error. And here it's the data set that we have seen so far, X and Y. In the clinical trials, in the setting that we focus, there's an intervention and binary variable. Let's say half people take the treatment, the purple drug, and the other half take the placebo. Together with Y, there are coming two other variables which is not fully observed. It's the Y1. What would have been the outcome if we were taking the treatment and we observed only for the half population, and the Y0? What would have been the outcome if we were in the placebo? And ultimately, we're interested for each person for the individualized treatment effect, that would have been y1 minus y0. And this, it's not observed, it's an unobserved variable. And this is then what we say the fundamental problem of causal inference, and you need to make some sort of inference towards this variable. So with our work, the objective was to identify a subset of x which is relevant with y1 minus y0, and having some mechanism that controls the type 1 error in these selections. So we took the knockoff framework, and we went step by step. For the step 1, we said that we can proceed with all of these methodologies that they are published. They create valid knockoffs for the problem that we have at hand. We needed, of course, to change the step 2 to identify knockoff statistics that now targeted the predictive strength and not the prognostic strength. And with the way that we generate the knockoff statistics by obeying the antisymmetric property, we're fulfilling also the third step. And this is the 2 knockoff statistics that we introduced, with sort of extensions of the lasso when we model interaction terms, and now we focus in the coefficients of the interaction terms. And when we use the importance score of the causal forest, which can be seen as an extension of the random forest, but this time targeting to estimate the conditional average treatment effect. And as I said, because of the way that we structure this knockoff statistics, we obey the antisymmetric property, and then all of the mechanism for thresholding them was the same as the original paper. And due to time, I will not show simulation results. I will just show how we use this mechanism in some real trials. In this case, we focus on psoriatic arthritis, which is an inflammatory disease, and it's quite nasty and affects a lot of parts of the body and the quality of life of the patients. And there is some treatment, blockbuster treatment by Novartis, the Cosentyx, which is indicated to active psoriatic arthritis for adults. We focus in these 4 phase 3 trials. And we wanted to identify predictive markers when we want to compare placebo versus the pool of the active arms. The primary endpoint, the very high level, the y, it's a binary variable, it's a classification problem. So y1 means that the person responded, y0 didn't respond. And we have 57 variables in the baseline. And now we want to see out of these 57, which one can be having, it's predictive, or they are effect modifiers, essentially. Now here, the estimate that we used was the causal risk difference, which is the probability of responding when you are in the drug minus the probability of responding when you are in placebo. And in the overall population of 1,600 patients, it was 0.26. We knew that the effect is very strong anyway. It's far away from 0, so it's a very effective drug. And then we apply the knockoff framework using the level of 20%, and we identify these 8 baseline characteristics as potential effect modifiers. And what we see here, in the dashed line, I have put the overall effect. Whatever sits to the left, it's worse than the overall. Whatever sits to the right, it's better than the overall. And the most promising variable, this C-reactive protein, defines some heterogeneity, because we can see that people with low values perform worse than the overall. Here, people with high values perform much better than the overall. So overall, a variable that defines heterogeneity is the one that moves, let's say, farther away from the dashed line. And we explore all of them. With age, we could see something similar. The younger population will respond better than the overall, while the older worse. And the deeper we go in this list, we see the variables starting converging towards the overall. And before finishing, I want to mention one paper that we had a few months ago that Manuela was leading this work, and he put some very nice work around the practical sides of this knockoff framework. And we tried to answer various questions. What I want to mention, which type 1 error measure to control, is very critical. So far we focus on the fault discovery rate, but by design, it has this characteristic because of this one in the numerator. No matter what are the truly relevant variables, the number of selected variables is also sort of lower bounded by the nominal level of 0.2. So when we have 0.2, the minimum selection will be or 0 or at least 5. So there is this gray area which will never have selection. So the method will never return 2, 3, or 4. So this is something that it's nice to keep in mind, especially in the subgroup discovery literature, that we assume that it's 1, 2, or 3 irrelevant variables only. While if you use this the other error, which is the per family, which is the expected number this time of false discoveries, we can see that we have selections across all of the numbers of select variables. So to wrap it up, the knockoffs provide a powerful framework for machine learning-based control variable selection. We focus in this presentation more on how to use the knockoff for identifying predictive biomarkers, and we have developed a tool, NER packets, the knockoff tools, that they can handle various scenarios, prognostic, predictive, various type of endpoints, even time to event, various different filter types, ones built upon regularized regression, random forest, causal forest, and various measures of error, of type 1 error. So yeah, thank you very much for attending. And
thank you, Kostas. Thank you very much. We're ahead of time, so there's plenty of time if anybody would like to ask questions, either using the chat or oh, excellent. We have somebody with a hand up already. Please.
You're muted, I think.
Yes.
Oh, sorry. I just make a mistake. I don't have a question.
No worries. Okay. Carl?
Yeah, hi, Costa. Thanks a lot. That was really nice. I enjoyed it very much. One question popped up when I was looking at your first couple of slides. You essentially change the properties of a machine learning model when you include all those knockoffs, right? And just my intuition is wondering, like, okay, to what? Sorry. To what to what degree does it change it, or how do you control for that? Because obviously, you worsen the problem of overfitting potentially and so forth, even though I know it's just noise variables, but somewhat it sort of strikes you odd. You sort of double your hypothesis space, right, with noise variables. So maybe you can comment on that a bit.
Yeah, this is a good question. In the work that we're doing, we're following this. We're doing this cross-validation, and actually in the lasso model, it was a bit more straightforward. In the causal forest, we didn't explore a large number of hypotheses, but I think maybe it will affect a bit the space of the hyperparameters. But it's true that with this, we don't guarantee that this set of variables will be the optimal, let's say, in terms of the minimum error of the lasso. It's more some set of variables that can be test this conditional independence that we had earlier, in terms of a Markov plugin, essentially. But it's true that, the models that we use, it wasn't very tricky to optimize. But if you use the models that are very sensitive in the optimization and doubling the dimensions even maybe can affect various other things, then this maybe it requires some caution.
Okay, thank you.
Okay, we have one hand raised and 2 questions in the chat. So I suggest, Steve, would you like to go first?
Yes, thank you very much. Costas, well done. I'm wondering, and I've thought about this for a long time, but never pursued it rigorously. I'm not familiar with the knockoff literature very well, but I've often thought of the reverse approach. Why not use the observed X's with all the correlation that's underlying all that, all the messiness that's in there, and then simulate Y's with known characteristics? That is, Y is 2 times X1 minus 4 times X7, blah, blah, blah. Or try different scenarios with 2, 3, 4 variable, etc. And then do that repeatedly and see how good your method, random forest, lasso, whatever, how well that performs at predicting or identifying the right 2 variables or 5 variables or whatever, and kind of reverse the process where I'm going to take the X's as they are, and I'm going to simulate known relationships with the Y, and then see how often my method correctly identifies them, and then, categorize or measure, like you say, how often did I get the right variable, etc. So I think that's a bit of a reverse of the knockoff approach. I'm just wondering if that's a reasonable thought or any reactions you might have to that.
Yeah.
Thank you.
Yes, thanks. No, I've never thought about it. Now, in this, the original Y, how do we use them? Sorry, I missed this part. We have also the original Y. You use them to infer some relationship and then simulate new Ys, or I'm not sure how.
No, I'm just saying yeah, you use simulated Ys to understand the operating characteristics. You know what the relationship is between the X's and the Y's, whether it's 2 or 4 or 10 or whatever. And you could try different relationships, linear, quadratic, interaction terms, and see what the operating characteristics are. And then when you're done and you analyze the real Y's, the one time you analyze the real Y's, you can see what your result is, and you have some idea of what the false discovery rate might actually be for the real data set that you're dealing with. Yeah. I don't know if that's a reasonable approach or thought, but anyway.
No, it sounds interesting, but I haven't thought about that. I need to put a bit more thought. Maybe we can have a follow-up. Thanks.
Okay, thank you.
We have time, or
we have plenty of time. There are a couple of questions in the chat, maybe related. Firstly, are there any assumptions required to ensure the validity of these knockoff procedures? And also, does the model performance get worse when you add knockoff variables?
Yeah, about actually, there are not assumptions a part of this one that I mentioned, how we should simulate the knockoffs. And so in theory, this is a method that can handle also indefinite sample size and correlations. Of course, in practice, when you have highly correlated variables, then at least what we're doing also, we do some sort of pre-processing step that we try to group the variables. But I want to emphasize that the following two presentations that we'll discuss a bit more in the methodology that they are tailored in the scenarios that we have highly correlated variables. And about the performance, to be honest, I never checked the performance of the models of the knockoffs, but I'm expecting that it will deteriorate. Yeah, the performance will fall when we use both the original and the knockoffs, but it's not of interest of us the performance of this high dimensional model. It's more the drivers, which is the importance scores. So at the end, we don't fit any model. You know, this methodology is not to produce a model, it's just to select the variables. And in the lasso regression example, did you use a group lasso? So the stringage of main effect interactions. Yeah, it's a good point. We didn't use any group lasso or some other methodology. So we allow the method to decide, you know, if it includes only the interaction or the main and the interaction. We tried to play a bit with that, but we saw that, you know, they didn't affect a lot. But definitely, maybe there's some way of improving more, that using some more fixed prognostic effect, and then just enforce always the prognostic inside the model, and then focusing the interaction with the predictive.
Yeah, I think we have.
Maybe I can try to answer them.
Yeah, we have a little bit of time, maybe.
Okay.
Yeah. But we're getting a lot of questions now.
Yeah. Yeah, for the sub values, there are works and that have used the global importance score. And actually in the paper, we mentioned one of them, but you can send me an email and can have a follow-up. How robust is the method to the knockoffs generation? What are the underlying assumptions to generate knockoffs? So actually, from my experience, let's say the generation of the knockoffs, if any, will influence a lot the power. So you want somehow to have some knockoffs to be as orthogonal as possible of the original variables, and this can lead, you know, this is a critical bit more towards the power and not so much around the type of error. Yeah, and there are various diagnostics around the quality of the knockoffs, and you know, one of the following presenters, Asher, has a great work around this. Do the methods apply beyond causal risk different to other measures of effect? The question of Gavin, I would say the causal forest filter targets only the causal risk difference. The regularized linear regression, the lasso, you can tailor it towards you can target other causal measures like the log-odds ratio, for example, in the binary case. So I think.
Yeah, thank you, Kostas.
Thank you. So Let me see. Dennis, are you in the audience? Let me see.
Can you hear me?
Yes, we can hear you. You can start sharing the screen, then I will introduce you.
All right, let's do that. I will also activate my camera. I won't be able to see you as I present my screen. I'm in a single screen setup.
No worries. We'll take the questions afterwards. If something, I will interrupt you. Yeah.
Okay.
So Dennis, we are very happy having Dennis. He's a biomarker and experimental medicine leader at Roche. He has a background in experimental psychology, neuroscience, and computer science. Before joining Roche, he pioneered machine learning approaches, bridging the gap between the laboratory and real-world applications of EEG and neuroimaging. At Roche, he's responsible for biomarker strategy and implementation in several drug development programs that support the neuroscience portfolio. Thanks a lot, Dennis, and the floor is yours, and we can see the screen nicely.
Thank you for anticipating my question and reading my mind. Yeah, really exciting to be here. Also really amazed by the broad interest by the audience, and wonderful introduction, Kostas. I think we can directly continue from here. I just want to give explicitly credits to Ahmad Chama and Bertrand Thirion, who are the two co-authors of the work that I will present today, related to two papers. And this is essentially the PhD thesis work by Ahmad Chama, who just graduated in Paris, a few months ago, 2 months ago. Okay. So I think I have a little bit of a delay. All right. Just to set a bit the context. So I think we share a lot of the context. So you can continue directly in your mindset from where Costa stopped. I think what is unique here is this particular perspective that I bring with from neuroscience, where we tap really into high-dimensional modalities like MRI and EEG, but also digital technologies alongside blood samples. And you have this setting where you have multiple modalities, and within those modalities, you can have structured correlations. And it's all a little bit more high-dimensional. And if you want to discover new biomarkers, you need methods that are fit for that purpose. And machine learning is an excellent framework because it allows for flexible function estimation with heterogeneous data. And in particular, it is particularly appealing to use the strengths of stochastic optimization to really build custom complex models where you can essentially layer by layer put in your prior knowledge that you have scientifically. And you can call this deep learning, but let's focus more on the utility here. However, if you want to move this forward in the context of research and development, you really want to have some statistical decision rules, because otherwise it is hard to move on and interpret the results of machine learning systems. Then as a small taxonomy, the approach now is that you look a little bit at the variable importance literature to move on. And as Costa nicely said, one approach is that you are model agnostic and you compare the performance of the model after you remove or permute a variable of interest. And this is where we are getting, but there are other methods too. So we saw some model-specific models, model-specific methods, but you also have local variable importance methods that tell you more about specific predictions at a single data point level, like Shapley values or Lime. And this is not what we are going to talk about. So we are here in global variable importance, and in particular here in this branch, in this model agnostic type of methods, and not the model-specific methods that we saw before, like Lasso, for example, specific methods in a causal forest. Okay, let's go back 1 second again to the context work. Ideally, what we would like to see is that we have a method that can really pick up all the relevant variables, while at the same time not picking up the irrelevant variables, and therefore controlling false discovery rate. And I feel in the statistical machine learning literature, a lot of work has been done on controlling statistical error control in terms of the false discovery rate or type 1 error. But you should also not underestimate in your domain-specific context the importance of having an adequate function estimator that represents well the function spaces that relate to your problem in your domain. Why this is important is clear. So we really need to detect the right variables. This is critical for discovery work, and it has consequences on studies that we plan or on variables that we include or don't include in study designs. Importantly, the focus on statistical guarantees is essential because commonly in the engineering deep learning literature that develops very complex and interesting models that are powerful, a lot of work is done through sensitivity analysis, and you have to endlessly analyze the properties of your system. And if you have statistical guarantees, that gives you an edge in liability management, and of course you want to have that, yeah. So you can save a few sensitivity analysis, hopefully. All right, now I want to focus maybe on the original or maybe most paradigmatic or classical or most famous model agnostic method, which is based on permutation importance by this seminal historic paper by Leo Breiman. The idea is that, we have X, we have Y, we are interested in the impact of this variable here on the prediction. So the idea would be that we permute J, and we then compare the performance of the model predicting Y before and after permuting. And this is typically done on the test set after we have fitted already the estimated the function. This method is known to lead to false positives and to spurious associations, and a lot has been said about this. I nevertheless want to stress that this method is alive and well and still further developed in contemporary applied machine learning research in biomarker discovery. For example, in this work by me and colleagues, 2021 in Nature Communications, where they integrated a permutation importance type of thinking into a fully connected type of multi-layer perceptron. So this is not a convolutional neural network. This is just any classical deep net that you can think of with several layers and classical nonlinearities. And the modifications that are interesting is that you're actually directly using the loss function, and you can get scores by comparing the loss before and after, which can save some time, obviously, because you get it from the model fitting. But also, you can actually then go ahead, and if you have a permutation distribution, you can average the permutation score that you get across your permutations, and you can calculate a standard deviation. And that gives you actually something that is a Wald type statistic, and it follows a t distribution or also a z distribution. And the authors here have actually used a z distribution with a normal approximation, and you get actually statistically valid p-values that seem to work nicely in the real world. However, we know that there is conceptually a problem. So the problem is that if you have correlations between the variables, things risk to break, and this is exactly what we proved in the first paper from Ahmad Chama's PhD thesis, which we presented at NeurIPS last year. And let's deep dive into this. So first of all, what's the alternative? We were quite impressed by this work by me et al. For the idea of using an expressive type of function estimator. But if you don't want to do permutations in the classical way, you want to have some conditional distribution from which you can sample. And you can achieve this in this type of manner. So what you do is that you are predicting from this notation here. So this is x minus j. So you're removing one column from the data set, and you're including it here, and you're learning a conditional expectation of j given the rest of the variables. Then you can predict xj, and you can permute essentially the residuals to compute this x tilde j. And this allows you then to sample from the conditional distribution. And now what we can do to speed up things a little bit, we are not necessarily forced to use a deep net or a very complex model for this inner business of learning the conditional distributions. And we found that a well-tuned random forest is actually just good enough for getting that business done. And this gives you a nice speed up where you actually have a conditional estimator that can be run in the same time as a normal non-conditional permutation approach, while still using a more heavy type of method. And notably, another type of method that's very popular is the leave one covariate out approach, for which you also have good statistical guarantees. However, it's important to really stress this is a refitting method where you have to refit your function for every variable of interest, and you introduce as additional variance, but also it leads to an explosion in computation time. All right, one nice thing that Ahmad did in this work is that he not only looked at the methods that we found most interesting, but we tried to really get together various methods that are used in the field that give statistical error control and that provide p-values. And in particular, we paid attention not only to the type 1 error, but also in the simulation setting to the capacity of ranking correctly the variables that is given by the AUC score, not to be confused with the performance metric AUC. So we are talking here about the recovery of the true coefficients and the correct ranking of the variables. And what you now can see, if you believe me, so these different points in different colors relate to different simulation scenarios. Our settings are 50 variables, 1,000 samples, correlation around 0.8. Some of these variables are predictive, others not. And what we vary is the type of nonlinear relationship where you can have only main effects, or main effects and interactions only from product terms, or more complicated nonlinearities with the ReLU function, or also a classification type of setting. And what you see here is that many of the methods, they are either quite good at controlling the type 1 error, or they are quite good at ranking the variables correctly, but you don't really see methods in the setting that do both well. To our own surprise, using the same DeepNet architecture as was proposed by me et al., we could reproduce the capacity of the model to detect the variables of interest, while at the same time benefiting from correct type 1 error control that is also performed by knockoff methods or loco, benefiting from theoretical guarantees. And what I don't show here, but what you can find in the paper, are proofs that show that asymptotically, once you have correlations in this type of method, the perm-fit DNN, you have non-vanishing terms that lead to distortions in your distributions when computing your z-score. So the cumulative distribution function is not correct any longer. And by doing conditional permutations, you're actually fixing it and all terms are vanishing. Yeah, and I think in the interest of time, I will skip this one. This is just a question of the asymptotic behavior. I think the only interesting thing that you can see here is that already in a smaller sample regime in our simulation, the conditional permutations are controlling the error in the expected way. I also would like to highlight once more this conceptual difference between leave one covariate out and CPI. Both are model agnostic methods that can be shown through statistical guarantees to control the type 1 error in the face of correlations between the variables. And this is what we see also here. However, loco is really a refit method. For fairness, we use the same base estimator here, the DeepNet. But you have to really refit everything for every variable. And this is where you get really faster with the approach that we proposed here. Okay, there are certain limits. So in some cases, variables are extremely correlated, especially if you think of images or certain types of gene data or omics. And you can even have super high correlations that are between 0.9 and 1. And then, of course, the classical way would be in a small data set that you look at the correlation matrix and you prune your variables. But if you do more data-driven discovery work, maybe you want to have a method that's immune here. Or you want to have an alternative, at least. Let's put it this way. I don't want to raise expectations too much. So if variables are extremely correlated up to a point that they are noisy copies of another, we run into a problem with the conditional permutation importance, because the information in one variable that we want to estimate is still present in the other. And so you won't really see a big change in your loss delta. You have mutual cancellation, and this is not working. So one way forward is to consider them jointly, either by using your prior knowledge on grouping or by including a certain clustering procedure or similarity procedure. Here we focused to make the point on grouping, because very often we want to combine images with other types of modalities, electrophysiological signals, and we just know that we want to have an inference that is limited to specific groups. And this is what we elaborated in the second paper that Ahmad presented earlier this year at the AAAI conference. So we are pretty much extending now the methodology from the previous NeurIPS work by introducing blockwise conditional permutation importance, where we say we have multiple blocks of variables that are user-defined. Otherwise, the procedure is pretty much the same. And this gives us blockwise p-values as before, pretty straightforward. And there are some interesting corner cases. So if your group size is 1, then you get just the classical single variable conditional permutation importance. And if the variables are uncorrelated, you fall back to the classical permutation importance. One important thing that I will also show here is that additional speed gains are possible through stacking procedures. Yeah, so this is a very classical idea in traditional machine learning pioneered by Walpert, where the idea is to have multiple models and you do cross-fitting, you take the predictions, you put them in a meta-learner, and you have a meta model that combines the predictions. What we did for a couple of years now in neuroscience, also in my previous work, was to, instead of having multiple models for the same data, have different models on different data sets. So let's say you have an MRI model, you have an EEG model, and you have a blood model. And you want to now get inference on the utility of these modalities. So the model is really specific to the modality, and otherwise it's the same stacking architecture. You have a meta model that then combines these modalities. And the trick here is that instead of doing this in a pipeline fashion, brute force, you can actually integrate this into your neural network through linear sublayers that perform something similar to PCA and create a subspace of an arbitrary dimension. To simplify things here, let's say we put it to 1. If you would have 3 inputs for different types of data or for different groups of variables, you would then compress them into 1 variable each. Okay, and again, we did broader benchmarks. We tried to incorporate as much as we were knowledgeable of in terms of the block-based group literature. And again, we see similar results. So you see here that the block-based conditional permutation method can deal very well with extreme correlation and at the same time control the type 1 error. And as a side note, you can, of course, use this with any other estimator. So you can also use this with a random forest or with a lasso model. It's model agnostic. And what you see here is that it doesn't work so nicely then in some cases. And again, you would see here that if you don't do conditional permutations and group-wise permutations, you get a problem in your type 1 error calibration when you're using just standard block-based permutations. There are, of course, also other methods like leap1 group out or leap1 group in that cannot provide p-values, but you can see at least in terms of the AUC score that some of them are better capable than others. In particular, leap1 group out, which is the analogous method to LOCO in getting a correct variable ranking. The stacking we found doesn't per se improve your statistical properties, so it doesn't lead to better error control or detection. But again, we can, in this case, speed up the fitting time by a factor of we can cut the fitting time in a half, which is nice. Okay, and before we close, I just want to give some real-world flavor, or some empirical flavor at least. What I show here is previous work performed at INRIA before I left INRIA a few years ago, where we analyzed different types of heterogeneous MRI and questionnaire and social and other types of data in the United Kingdom Biobank in a subset of 8,000 participants to better understand the relationship between those variables and aging, cognitive functioning, and mood, but also additional health variables related to lifestyle, such as exercise, sleep, and smoking. And in one subset of these analysis, we performed essentially a leave one group in informal variable importance analysis that would give us some idea about the importance of brain imaging, for example, for age prediction or for prediction of cognitive scores. But we really didn't have a fine-grained inference. We didn't have p-values, and you could hear, for example, this is without brain, this is with brain, and you would see that there is a big difference. So you can predict the age of a person from all sorts of epidemiological data. But once you have the MRI, it's getting much better. But this is limited. And revisiting this particular example with the methods developed in Ahmad's thesis, we obtained far more fine-grained inferences that could show us that for the case of age prediction, for example, lifestyle shared non-redundant information with the MRI and also with education. However, functional MRI using the Bolt signal or other types of mental health measures did not contribute to the prediction here. And while this is not the primary intended use case, you can, of course, use an approach like this also for variable selection and reduction. And of course, you have to do this in a careful way with cross-fitting, not to have spurious results. And what you can see here is that if we filter at different significance threshold and use a reduced model, we preserve the prediction score in this context. And this can also be useful. So you have a clear road forward for variable selection as well. Last not least, there's an exciting new package that is currently nascent, developed by the colleagues at INRIA. It's called HIDIMStat, and it's becoming a home for high-dimensional statistical inference algorithms in the context of machine learning. And we have also now contributed our methods here. You find everything in one class, the block-based importance. It's compatible with scikit-learn. And by configuration, you can choose the estimator that you want. It doesn't have to be a deepnet. It can be an SVM. It can also be a random forest or what have you. And you can implement all the flavors of algorithms that I just presented. There's also R code that we wrote to give an example of how to do conditional permutation importance with random forest, for example, but there is no package yet, so we are working on that. To wrap up, I presented to you conditional permutation importance methods that are model agnostic and that capitalize on using statistical guarantees. We saw that this allows us in particular to explore more expressive base learners while getting some computation time saving in using a more approximative, faster method for the conditional importance estimation. And this performs well even in the face of correlated variables and is backed by statistical guarantees, and importantly faster than local methods. This is then extended by block-based permutations that allows you to explore these methods in high-dimensional structured data sets with extreme correlations. And once more, this is really flexible, so you can really plug your own models here. You don't have to use our models. You can use the model that does a good job at empirical risk minimization in your prediction problem. And that's it. Thank you.
Thank you very much, Dennis. Are there any questions? Maybe I can start. Let me ask. There is one in the chat. So, is the high DME library also planned to be implemented in R?
No, I think not, because I come from a Python environment, and INRIA is also that lab, really a Python place. But of course, we would be happy to facilitate such an effort. I mean, I think at least I'm very interested to also provide an essential R code, but I think it's just not prioritized at this point. But I mean, if there is interest, it sounds like a good open source project.
Aditya has one question. Now I can read it, but if you want to unmute the distill condition randomization term D0CRT in your earlier power plots also controlled type 1 error and had very good power, except when there are only interaction, which is perhaps expected. Any comments on your comparison to that method?
Yeah, I mean, this is a method that was also studied at INRIA, and I think it is just in that case not as expressive. So I think the function space this method is dealing with is just more limited. So I think it's really about the nonlinearity.
One question from my side about the grouping part. So you assume that there is some known grouping of the variables, or it's also, or you learn some grouping from the data with some way?
Yeah, I mean, this is really up to the researcher. You should, yeah, I mean, the method doesn't do anything. It requires that you provide group definitions. In our case, working with multimodal data, or sometimes also working with multiple types of recordings. And I mean, let's say you have different EEG recordings, and some of those EEG recordings are done yeah, at different times of the day, or yeah, with different types of medications. You have a group that is given by the data acquisition or the context, or you would have imaging versus another group. And it's very intuitive there that you do not necessarily want to mix those variables because the data generating processes aren't connected at all. Yeah. And this is where I think also sometimes, yeah, you are just not having a scientific question to the single variable that is in your data set. So in EEG, for example, you're interested in learning more about frequency bands, and you don't necessarily always care about specific electrodes that matter. And then what you can do with this grouping approach is that you get inference at the granularity that you are interested in scientifically without necessarily needing to do ad hoc feature processing or preparation. I hope that makes sense.
Yeah. So the implementation now it requires some grouping.
It requires grouping, yes, that come from your knowledge or that you could also obtain through another procedure. And as I said, the methods are scikit-learn compatible estimators, and you could of course include some clustering method, or you could do clustering on a training data set or somewhere else and use data to define groups.
Yeah, makes perfect sense. Yeah. Any other questions? Carl?
Yeah, thanks. Maybe a quick one. Thanks again for this intriguing talk. I remember at some point there was this conditional variable importance introduced by Karoline and Strobel for random forest. That's quite some time ago. And I was just wondering, how does it sort of relate to what you are doing here? If you can like tap into this a bit, that would be helpful to differentiate the two things if they're different. I mean.
Yeah, absolutely. No, this is really a good question. We studied this work closely, and actually we benchmarked it also in the NeurIPS paper. So this is one of the things that we looked at. There was one result on the conditional random forest that in the context of this simulation had medium performance. And I think the context is just that I think that work was really seminal and eye-opening, and in a way teaching us how to think about the problem. It was, however, really closely related to the random forest land, and the intuition is coming from the more high-dimensional type of data. This is not what really works in that setting. So yeah, I think this is perfectly fine if you have tabular data and you are in this classical survey land where you have very long data frames with a few dozens of variables maybe. And I think this is just fine. And I think in a way, what we try to do here is extract those insights and try to generalize them. Yeah, but the Strobel work and the Strobel and colleagues' work actually was part of the benchmarks. You find it in the NeurIPS paper.
Okay, yeah, thank you.
Thank you very much once again, Dennis. And maybe we can move on to the next presentation. Yeah. Aser? Great. So thanks a lot. I will introduce you. And Aser is a fourth-year PhD student in statistics in Stanford University, where he's advised by Emmanuel Candes. Aser studies problems in high-dimensional statistics, statistical machine learning with application across disciplines, including genetic studies, financial, economic applications, exploratory analysis of clinical trial data, and many more. Previously, Aser was undergraduate at Harvard, where he was advised by Lucas. And I will especially want to thank him because he's in the West Coast for waking up so early to join this. Thanks, Aser. But now we cannot hear you for some reason. Still no.
Okay, what about now? Is this better?
Now it's great. Thanks a lot. Yeah.
You're good? Okay. Okay, well, so first of all, thanks so much to the organizers of this wonderful seminar, Kostas, Dominic, Trey. It's really great to be here. I'm going to try to share screen. Let me know if this, can you see this?
It's coming. Yes, it's great. It's great.
Okay, fantastic. Let me just set up one thing. Sorry, so I can see you at the same time. All right, and let's get started. So thanks so much, everyone. As Kostas introduced, my name is Asher. I'm a rising fourth-year PhD at Stanford University, advised by Emmanuel Candes. And today what I'll talk about is controlled discovery and localization of signals via a method called Bayesian linear programming, or BLIP for short. Before I get started, I just want to thank my really spectacular collaborator, Lucas Jansen at Harvard. I've learned a great deal working with him. It's been a lot of fun. So if you ever get the chance to do it, I highly recommend it. Okay, so I thought maybe it'd be helpful to start with a sort of a one-slide overview of what I'm going to talk about today. So, you know, I think the challenge, as we actually heard in the previous talk, is that sometimes when we're performing high-dimensional variable selection, there are correlations among the variables that make it challenging to identify which variables are important. So, you know, often we can tell that some variables influence the outcome y because, you know, our model is predicting well overall, but we're not really sure which ones because, you know, the variables are highly correlated with each other. And, you know, even after fitting a model, it's sort of unclear how to localize which variables may affect y. And maybe just to give a little cartoon of what I mean by this. So, you know, imagine we have 5 variables, x1 through x5, which I've shown in different colors, like maybe because they're different modalities, like maybe some is image data, some is electrophysiological data, as we heard before. And then we have some outcome y. And you can imagine maybe the blue variables are correlated and the green variables are also correlated. So, you know, maybe we don't have enough power to detect that x1, for example, is actually influencing y, but we can tell that either x1 or x2 is influencing y. And maybe we also have enough power, even though x4 and x5 are correlated, to just know that x5 is the true signal variable. x5 is actually what's predicting y. I mean, so that's the kind of inference I'd like to do today, right? And basically, I'll sort of make 2 notes here. First, I didn't sort of pre-specify in advance any groups. I didn't have to like partition my variables in any way in advance. I sort of just automatically found the smallest set of variables as possible, where, you know, in each group of variables I select, so inside each of these red circles, there's at least one important variable. There's at least one variable that's genuinely predictable. And you know, some conditional sense. So, you know, what I'm going to talk about today is how can we do this? How can we get these types of red circles? And this talk is a little bit of a strange talk because I'm not going to tell you anything about how to fit a model today, or which model to fit, or, you know, a frequentist inference procedure, really. Rather, what I'm going to talk about is how to extract useful information from a pre-fit model. So, you know, what in today's talk, I'm going to assume that this model is Bayesian. So, you know, you can imagine fitting your favorite, you know, Bayesian linear regression method or high-dimensional Bayesian regression, whatever. I'm just going to ask, how can we extract as much useful inference as possible? And although this is perhaps a slightly non-conventional subject, I just think empirically this matters. So what I'll show you is a large-scale genome-wide association study where this approach increases power 20 to 50% compared to a previously published result with less than 1 minute of added computation. So there is really some benefit to doing this type of extraction and thinking carefully about what, you know, types of inference we'd like to perform, and then trying to do it as well as possible given a model that we have. So, what am I going to talk about sort of more specifically? I'll give a motivation. I'll talk about the core methodology, blip. I'll give an application to genetic fine mapping. I'll then give sort of a quick advertisement for this new work, which just as a disclaimer, I'm not a co-author on. I just think it's really cool, which is sort of an alternative knockoff space method that sort of accomplishes a similar task in a frequentist way. So I'll sort of talk about that for one slide, and then I'll conclude. OK, motivation. So the main data set I'll talk about in today's talk is a UK Biobank data set where Y is disease status. I'll look at a couple of different Ys, but sort of a running example will be cardiovascular disease. X1 through XP are genetic variants, and there's 19 million of them. So this is a pretty high dimensional data set. And the question that I'd like to ask is which features, which mutations XJ influence Y? And if XJ influences Y, I'll call it a signal. So, as sort of just to get ideas down, for simplicity, let's temporarily assume a linear model. So I'll write that the outcome Y equals X transpose beta plus some noise epsilon, where epsilon is mean 0. And I guess I'll define a signal throughout this talk as just a variable which influences the conditional mean of Y given X. So in this linear model, that's just variables with non-zero regression coefficients. So to be more precise about the challenge, in this particular genetic data set, the mutations exhibit strong local correlations. So for example, the correlation between X1 and X2 could easily be 99.9%, right? And to be clear, these are different genetic mutations. Like, they may have different scientific meanings. Like it might be that one of these mutations is actually causing a disease and the other one is not causing it, right? They're just correlated because they're nearby. So we do genuinely care about finding the mutations which cause a disease of interest, but we may have no power to detect that, for example, beta 1 is not equal to 0 because X1 is too correlated with X2. So instead, as we saw sort of in the previous talk, maybe we can tell that X1, X2 together contains a signal. So just to give you a little illustration of what this looks like, I think this is a cartoon plot. This is not based on real data, where the x-axis is sort of showing 100 different mutations, and the y-axis is showing a log p-value that's essentially a measure of the correlation between y and x. And what you see are sort of these smooth peaks that are spatially, smooth in some sense, because there's sort of local correlations among mutations on the genome. So, when we see a picture like this, like it's not immediately obvious where the important mutations are, where the causal SNPs are, for example. Like, clearly there's some peaks around like the location 15 and the location like 45 and the location 80 maybe, but it's not completely clear where exactly the signal variables are. I mean, I can tell you because I generated this data myself, there are 5 signal variables. They're shown by the red X's. But if you fit a linear model, you might find that nothing is significant. So the question is, how do we avoid this? So, this motivation, I think, is not just specific to this particular data set, as we also heard from Dennis a little bit. Correlation and correlated features really do appear in exploratory analysis of clinical trials. So this might happen if you have, omic data like proteomic or genomic data. But it can also even happen when you just have demographic data like age and baseline variables. It's not uncommon for different baselines to be correlated with each other, I think. And throughout, by the way, I'm just going to think of this set of features X as a genetic set of features. So if you have a clinical trial, this could include treatments and interaction terms and so on. So yeah, just as a little bit more motivation, I've just taken correlation matrices of some selected set of covariates from 2 clinical trials with publicly available data. I believe the left trial is from Bloom et al. Is a trial evaluating methods for treating postpartum hemorrhage, and the right trial is an HIV resistance testing trial. But you can just see that these correlation matrices have actually some reasonably strong correlations, I think, because baseline and demographic variables are not unsurprisingly somewhat correlated. So if we want to know what's truly modifying a treatment, if we want to know what's truly prognostic, we sort of may have to deal with correlations. And of course, this is particularly true if you have medium to small size data sets. Then even modest correlations are going to make it much more challenging to identify important variables. Okay, so what's the contribution? I'm going to show a method which discovers disjoint groups of variables, G1 through GR, which each contain a signal, and they contain a predictive variable. Our goal is to make R large and G1 through GR small. So what I mean by this is that we want to make as many discoveries as possible, but we want each discovery to be as small as possible because we only know that there's at least one important feature in each group. So we sort of like to narrow that as much as possible so we can isolate which features are important. And I just want to emphasize these groups are not, pre-specified. You don't have to run any clustering yourself. You don't have to do any pre-processing. These groups are just automatically output by the method. And then lastly, this method will control the Bayesian false discovery rate. And in particular, as an input, it takes posterior samples from any Bayesian model. So, this could be a Bayesian GLM or a generalized additive model. And then it outputs these groups that sort of maximize this notion of power I've described subject to FDR control. But the asterisk for today's talk is that this FDR control result assumes that the Bayesian model is well specified. So you need to believe in your model. Like this method is not going to work, or is not going to give false positive control if your model is completely inaccurate, for example. Okay. And just to show what this looks like in a picture, so if we sort of go back to this cartoon plot of a partial Manhattan plot, you can see that if I just sort of give inputs from a sparse Bayesian linear model in this setting, what it produces is outputs that look like this. These sort of green bands where each band is a detection region. And we say that each region contains at least one mutation that is impacting the disease. So for some peaks, for example, the 2 peaks on the right, these bands are really narrow. They just contain one mutation. We're just completely sure, or we're very sure that those mutations are the ones that matter in predicting or causing a disease. And then some bands are sort of naturally more wide because there's more correlation among the variables. So for example, this band around location 20 probably contains like 5 or 6 different mutations. We know that there's some relevant mutation in that region, but we don't know exactly which one it is. So that's sort of the type of method that's the output of the method I'll show you today. Okay, so now we'll talk about methodology. So as notation, X are going to be features, Y is an outcome, and script D is the data set. So I'm just going to assume first that the analyst has specified a Bayesian model, which implies that the expectation of Y given X is equal to some function f theta of X for some parameter theta in capital theta. I mean, this could be a very large non-parametric model. Theta could be infinite dimensional, or it could be a linear model, which is what I'll actually use in the real application. And theta is sampled from some prior distribution, so this is truly a Bayesian model. And then I'll just call this set of signal variables S to be the set of variables J, such that the conditional mean f theta of X depends on Xj. So then the second assumption I need is that you can sample from the posterior distribution of the parameter given the data set. So basically, you can fit this Bayesian model through MCMC or variational inference or something like that. Of course, there's a huge Bayesian literature on these types of sampling techniques. This problem is not straightforward, but I'm going to assume that you can accomplish this. Again, this assumption is not always reasonable. There are some models for which this is computationally intractable. Sometimes you might not have prior information, but sort of the scope of this talk is one in which these assumptions are true. And then the question is, just how do we extract useful insights from these models after fitting them? Okay, so our goal is to discover disjoint groups which each contain a signal, and discover as many groups as possible and make them as small as possible while controlling the false discovery rate. Again, I do want to emphasize one final time that we'd like to pick these groups after seeing the data. So one simple example is you might see a correlation matrix that looks like this. We have like age, sex, blood pressure, and 3 biomarkers. Like this is sort of a toy example where the correlations are pretty high. But if I asked you to make groups a priori before seeing the data, it's not clear, for example, if you would group all the biomarkers together, if you would put them into different groups, if you include age in that group or not. Like it's just not clear how to choose if you're going to apply some clustering algorithm, it's not clear how to choose the cutoff for the hierarchical clustering, for example, because it depends on how strong the signal size is. So we'd like to only do this after seeing the data. So basically, what I'm going to try to do is define a method that maximizes power subject to controlling FDR, and such that these discovered regions are disjoint, so that we don't have any redundant discoveries. But I guess the first question that we need to answer to actually solve this optimization problem is just to ask, what mathematically does high power look like? I mean, so as we've discussed, we want to make as many discoveries as possible. We want these discoveries to be small. But I think in existing work, there's not too much formalization of what power means. So it's sort of hard to directly optimize it. So what we're going to suggest is basically defining a weighting function, W of G, that measures the value of discovering a group. And this can be specified by the analyst, but it can also, we'll give a default choice in a second. basically the idea is that just W of G should penalize larger groups because discovering a larger group contains strictly less information than discovering a smaller group, right? All you get is a guarantee that there's at least one feature somewhere in the group. So larger groups are less useful. And sort of a canonical choice that we suggest is just using 1 over the size of the group. So, roughly this means that if I tell you that one of 2 mutations is significant, but we're not sure which one, that's half as valuable as just precisely isolating which mutation is impacting the outcome. Again, you can, if you think that weighting is not suitable for your application, you can choose your own weighting. And then we're just going to define power as the sum of the weights of true rejections. So this is just the sum over all R rejections, where IGR is the indicator that G contains a signal, and WGR is just the value or utility of discovering the group. So this has a couple advantages. I mean, using different weights can accommodate very different scientific objectives, which is good. Sorry, that should say objectives, not objections. In practice, we don't always know exactly what our utility function is, but as I'll show you later, that the results are not too sensitive to the precise specification. Like if you take the log of 1 over G, it doesn't really make too much of a difference compared to just using 1 over the size of G. So if you have a completely different objective, then you can get different results, but the results are not too sensitive to this definition. We tend to find it. Okay, so what is the method in this talk? The method is just to directly solve this optimization problem. We've now sort of defined power. We have sort of some notion of false discovery rate that Kostas talked about, and we want to ensure that the discoveries are not redundant, that they're disjoint. We're just going to directly solve this optimization problem. And sort of the key theoretical result is that this optimization problem is equivalent to a very large integer linear program. So in particular, if we let ZG be just an indicator variable that we discover a group G, then it turns out that both the objective power and the constraints, false discovery control, and having non-redundant discoveries are just all linear functions of these indicator variables. And here, just as notation, the sort of key quantity that we need the Bayesian model to provide us with is just the probability that any group G contains a signal variable, and that sort of can be computed under the assumptions I discussed earlier. So this is progress. I mean, now we have sort of a name for this type of problem, right? It's an integer linear program. Those are fairly well studied. But the problem is that we have 2 to the p integer decision variables, where in our application p is 19 million. So that's clearly too large of an optimization problem. So we have to figure out a way to solve it. So basically what we're going to do is we're going to narrow the search space after looking at the data. So basically the first thing we propose in our paper is just to discard these groups which have a very low chance of containing a signal. So if something has less than like a 0.1% chance of containing a signal, you just don't need to include it in the optimization analysis. You can just discard it. That already in sparse problems reduces the problem enormously. The second thing we do is we narrow the search space by imposing sort of desirable structural constraints. So for example, we are unwilling to discover a group with more than 25 mutations in our empirical analysis, just because we think maybe a region with more than 25 variables is too large and it's not sufficiently informative. Again, you can pick your own structural constraints for the relevant problem, but this helps us narrow the search space. And then lastly, once we do this, this actually reduces the problem to a reasonably manageable size in a lot of cases, in which case we solve the continuous relaxed version of the linear program and then round to obtain integers. And what I'll just say quickly in this plot is that, I mean, this gives you provable FDR control. It doesn't always solve the optimization problem exactly, but in some simulations, we find that the expected power versus a provable upper bound on the power are these blue and red lines, and they're essentially indistinguishable. So, these sort of common sense heuristics do pretty well in solving the problem. So that's basically it. That's the method. What we basically did is we just defined a notion of power, and then we just directly optimized it subject to false discovery rate control. So as an input, all you need is a Bayesian model and sort of any desired structural constraints on the discovery set. As an output, you get disjoint discoveries, which verifiably nearly maximize power and control the false discovery rate. And I'll just show you here that here's some high dimensional variable selection problems where p is the dimension and n is equal to p over 2. And I basically fed 2 different models. The blue line is a linear spike and slab Bayesian model, and the red line is SUSY, which is a Bayesian model we'll talk about in a second. And what you can basically see is that the cost of running this post-processing procedure blip is very small compared to the cost of fitting the Bayesian model. I mean, it essentially requires solving a very small linear program, so it runs practically instantly. Of course, fitting a Bayesian model in high dimensions is expensive, and we make no contribution to that. I can't tell you, I can't give you any more guidance on that than the literature already does. But at least once you've fit a Bayesian model, I think it's valuable to run this post-processing step. Okay, so let me sort of prove that to you in the application. So just as a recap, the data set has 330,007 observations and 19 million SNPs, mutations. There are 4 traits of interest. The Bayesian model we're going to use is SUSY. It's a sparse Bayesian linear model that can be fit highly efficiently. Like BLIP, it actually returns regions of the genome where each region contains at least one signal variable. But these regions are sort of constructed heuristically. So today we're going to ask if we can do better using a principled approach. And we're just going to run BLIP directly on top of a prefit SUSY model that was published in another paper by Wasbrod et al. In 2019. So again, I'm not going to fit a new Bayesian model. I'm just going to run our post-processing procedure directly on top of the original Bayesian model. So the results are basically that I defined a metric of power, where I sort of measure the sum of 1 over the group size. And you can see by this metric of power that I have defined, our method, BLIP, which takes 4 minutes to run on this whole data set, increases power by between 30 and 50% on these 4 traits. So we make, we make more discoveries, we make discoveries at finer resolutions. This is good. But I think you might also reasonably be skeptical that this is a notion of power I have suggested. So perhaps it's not shocking that my method does better on this sort of optimality metric that I myself have introduced. So I guess the second plot I'll show you is I'll show you the cumulative frequency of the discovered group sizes. So again, each here group contains at least one signal mutation, with a subject to false positive control. And you can see that for every size k, SUSY plus BLIP makes more discoveries than SUSY does originally. So whatever, as long as you agree with me that sort of narrowly isolating, causal mutations is of interest, I think by any measure, this method really improves our ability to sort of narrowly isolate these causal mutations. You might ask also if our results are real. So we sort of performed a corroboration analysis where we checked which discoveries can be corroborated by a separate study in basically a large-scale GWAS catalog. So I'll sort of, if you look at the left-hand column, this is the corroboration rate of the original study. And the right-hand column is the corroboration rate of just the new discoveries made by BLIP. So it's not the discoveries in common, it's just the extra discoveries we made. And you can see that the corroboration rates are somewhat comparable. I mean, they're a little bit lower on the right-hand side. For example, in the cardiovascular case, this is the worst case for us, where the original corroboration rate is above 82%, and then ours is about 65%. But I think, you can interpret these results how you'd like. But my interpretation is that this is actually a reasonably positive result. Since sort of all of the low-hanging fruit, all of the completely obvious known mutations should appear in the left-hand column. So I think it's actually quite remarkable that these sort of new discoveries we made have a corroboration rate that's even comparable to sort of the left-hand column, which sort of contain all the low-hanging fruit. Okay, I'm running out of time, but I only have really one more slide. So one possible weakness of our method is that we assume the Bayesian model is well specified. I mean, if you really believe in your Bayesian model, this is a strength because it will grant you more power. But if you're not completely sure about your prior, this is a little bit of a weakness. So I sort of thought I'd just give a little advertisement for this really excellent work from Paul Gablantz and Kara Sabati in my department, which solves sort of the same BLIP optimization problem, but obtain model-free frequentist FDR guarantees. So, I mean, this is nice. The insights are that they use knockoffs for model-free error control, like Kostas talked about. And then they sort of have the technical insight, which is to use e-values to account for multiplicity. And I think maybe Aditya Ramdas will talk a little bit about e-values later. But ultimately, the takeaway from this slide is look, the problem I have suggested, I have a Bayesian solution to it, but I think the problem statement is what really matters. And that problem statement is not Bayesian or frequentist. It's just a question of how can we extract useful inferences from models in variable selection problems where we have correlated features. Okay, so that's it. So I introduced BLIP today, which is a powerful and efficient method for resolution adapted variable selection. It has provable Bayesian error control and verifiable near optimality in the sense that it sort of maximizes this notion of power I've introduced. It empirically gives a lot of power gains on a fine mapping application. There are both Python and R packages available to use this if you're interested. There's a lot more in the paper, including applications to astronomy and change point detection, potential for other signal discovery problems with spatial structure. And I think I'll leave it at that. I'll just say the paper is available. All code is posted. And thank you so much, everyone, for your attention.
Thank you very much. So, are there any questions? Yeah. Do you want to unmute yourself, Cetera? Fair. Let me read the name again. Yeah, Cetera. Do you want to unmute? Okay, it was a mistake. One, yeah, from my side, also I want to point out the paper. You have some very nice applications, or also around the time series change point detection of this method. One, a bit more. Okay, we have more questions. Let me go here. Cornelia.
Actually, I have the same question that looks very interesting. Application to astronomy. If maybe just a few words could be said about this.
Yeah, for sure. I mean, so I guess, let me just sort of go back a bunch of slides. So I have some. Let's go here. So, I mean, sort of the overall philosophy of this problem, right, is that there's some signals somewhere. We don't know exactly where we are, but we can localize them to some region, right, of the genome. And I think that structure arises in a lot of problems. So like Kostas was mentioning, it does arise in change point detection. Like often you can see that a time series has changed over time, but you don't know exactly where it has changed. And it also arises in astronomy. So like one application we do in astronomy is that what one subject is sort of identifying exactly where a point source or a star is in the sky. And that because there's sort of blur in telescopes, you're sometimes a little bit uncertain about exactly where that point source is. And sort of what our method can do, I mean, it's actually not too much of an extension at all of the mathematics, is just to sort of define sort of circles or ellipses or other regions where the circles are sort of as small as possible, and each circle contains at least one point source or star, something like that. So it's again sort of a spatial problem where we're trying to narrow down as much as possible where signals of interest are. In this case, the signals are stars. And we have an application where it's sort of the ground truth is available because like a much more powerful telescope looked at the region later. And you can see that our method improves power a lot while actually yielding almost exact FDR control. It's sort of remarkable that the FDR level, the actual achieved false discovery proportion is close to Q for lots of different values of Q. So yeah, the astronomical application is cool.
Thank you.
Yeah, thank you.
Any, maybe from my side again, because all of these applications look very exciting and really nice in a high dimensional setting. Do you feel that this method can be also useful when we work a bit more to the clinical trials? Remember with this low dimensional setting of 50, 60 variables and, I mean,
I think so. Yeah, I mean, so I think genetics is sort of a natural example for us because there's sort of it's sort of more canonical to think about regions. Like, people have talk sort of concretely about like, oh, let's identify which regions have significant mutations and then sort of narrow them down as much as possible. But I think like I was mentioning here, like, okay, this is a toy example, but like you could imagine a correlation matrix that looks like this, or maybe has a little bit weaker correlations. But if you only have like 100 data points, it's not going to be super easy to distinguish whether biomarker 1, 2, or 3 are going to be treatment effect modifiers, or whether they're going to be prognostic variables or something like that, right? Like you might genuinely care about which of those biomarkers is affecting the treatment effect, but you might not be able to tell. So that's sort of where this method comes in. It sort of tries to narrow which biomarkers are relevant as much as possible using the data at hand.
Amazing. And of course, you can handle mixed data types and all of that by the underlying Bayesian model.
Yeah, that's right. We make no contribution towards that. I mean, we hope you have a Bayesian model which can handle those mixed data types, and then you can sort of wrap our method on top of it. But
yeah, good. And any other questions? So if there are not, no, thanks a lot, Arthur, and thanks all of the speakers. Yeah, we finished a bit earlier the first part. Now we can have a break around 10 minutes it is. Okay, let me check the agenda. Yeah, 10 minutes. Oh, so we didn't finish earlier. 10 minutes break. And then, yeah, we'll see you in 10 minutes. Yeah. Thanks a lot.